Yeah. Can folks hear us on Zoom?
Yeah.
Cool. Thank you. Everybody's all months ago. Megan, do you know how to get us to your screen and showing our photos too? Yeah. Or does it? Yeah.
I don't know about the rain, but there's.
Oh, here you go. Okay, so you have to just leave that there. Yeah, okay, just cross the finishing line, is something you wanted to be? Oh, let me leave it. I'm sorry, it's okay. Yeah, right. Yep, and I'm just like double checking everybody. Sounds good. Hello. Hey, how you been? I love it. Are you? Yes, yeah, I will take a class. So, thank goodness. But yeah, I'm so like. Yeah. How are you guys doing? She's been texting you. Shows up at a lot of meetings. Yeah. Yeah. So yes. Total restoration, right? Yeah. Oh.
Welcome everybody to Megan's Defense. It's great to see such a huge audience on the week after exam week. So thank you everybody for sticking around. And we have some special guests from our lab from past years too. So it's really fun to see some familiar faces. But I do want to give a brief introduction to Megan for those of you that don't know her or maybe are less familiar with her past or maybe some of her accomplishments while at USU. So she started as a master's student in our lab in August 2022. However, she did a lot of things before she got here. She packed a lot in to just a few short years. So a really pivotal experience for her was going to Greenfield Community College, and she graduated in 2018 from there with a degree in environmental conservation. And I understand that the faculty member from there who first got her into research is on Zoom today. So that's pretty exciting to hear. From there, she was also a RU researcher at the Harvard Forest in 2018. I had to do a PowerPoint because I really could not keep all of these details straight. Holy cow. She graduated from UMass Amherst in 2020 with an environmental science degree and a really impressive honors thesis. I'll comment on in just a second here. She also headed out then to become a monarch habitat field tech in 2020 with the Monarch joint venture in Minneapolis. She kept heading west and served as a AmeriCorps volunteer in 2021, was a community science educator in Port Townsend, a lovely place in Washington State. And from there, headed to a River Watershed Association where she was environmental science and engineering fellow. So lots of really interesting things on her resume. But I received her resume and I thought, oh, she sounds like such a lovely person. And then I had the chance to talk to some of her references. And, sometimes these conversations really move you. You realize, wow, you've got somebody special here. So some of the things her references said, Megan creates space around her that other people wanted to join. I thought, this is somebody I want in my lab, absolutely. She's not arrogant at all. She just has a presence. She carries the room in just a really lovely way. She's passionate about education and teaching, which was really an essential part of her project and her role in our lab as well. Her honors thesis was huge. She figured out how to do everything. So she had clearly a lot of independence and just ability to figure a lot of things out. She's extremely professional, carries herself like a graduate student. Sign me up. So it's been a pleasure having Megan in our lab. She's really accomplished a lot of things. So I have a few awards for her I would like to share with you. Definitely, she gets the award for being most game for anything. Do you want to give a pub talk in Salt Lake City for the Society of Wetland Scientists? Sure. Okay. Would you like to spend your Sunday morning on an airboat ride with the dean and show her all about aquatic plants? Absolutely. Megan signed herself up. How do you feel about developing lectures and quite a few labs on aquatic plants and habitats for all of my classes? Absolutely. And she, I'll just say that the students here at USU were truly lucky to have her in the classroom quite a few times over the last 2.25 years here. She also is incredibly artistic, and you'll see that with her slides here today. Look at this lovely painting from one of our lab events. I might actually say, though, she might be our most improved artist. This was our lab pumpkin painting event. I'm not sure what the stripes are about. She also gets the award for the best gift to a lab member. I still don't know why we don't all have these sweatpants. This is mostly for Megan's benefit. A gift to Callie, her tech the first summer. We're the World Lab, if you couldn't figure that out. Also, most excellent mentor. She has, we've had a couple undergraduates who did research with her, but Megan has also mentored quite a few people in our lab over a brief time, and I think everybody has benefited from that. She also gets the award for most organized and elaborate plan to complete her thesis on time and put her advisor spreadsheets to shame. So we have been having weekly, if not twice a week meetings, and Megan shows up with her spreadsheet, checks off all of her progress, lets me know what she's going to do in the next week, and then delivers that and everything else and everything more. So really impressive. And then finally, Megan's experiment, one of the experiments you'll hear about today, the award for the most logistically complicated and risky experiment that caused her advisor to question her own her own judgment. So you'll hear about this really interesting, amazing experiment with June suckers. And holy cow, I didn't think that.
I don't think you were the only one who didn't think it was.
We were both not sure if she was going to be able to pull it off, but she absolutely did. And you'll get to hear about those results today with her research and quite a bit other really amazing research that she's been doing on an aquatic plant. So I will turn that over. I'll turn this over to Megan here and let her tell you all about what she's been up to the last 2.5 years. Oh, yes, I was about to walk away.
Okay, thank you, Karin, so much for that introduction. That was very sweet. Today I'm going to be talking about my master's research, which is focused on Potamogeton crispus, or curly leaf pondweed. That's an invasive aquatic plant, and we'll be talking a little bit about its impacts on our native aquatic plant communities, but also those lovely June sucker that Karin just talked about. In this presentation, there's going to be 5 main sections. We'll start with my research motivations, go into our study system that we use to answer our questions, and then 3 and 4 are going to be about my 2 research chapters that are in my thesis. The first one looking at those interactions between curly leaf pondweed and our native aquatic plants, and the second one looking at the potential impacts of curly leaf pondweed invasion on June sucker habitat. And then, of course, what is science if we don't offer actual management implications from it? So we'll end with some considerations for future aquatic plant management. Okay, but before we get into the science and the jargon, I just wanted to take a second and show everyone one of my favorite photos from the past 2.5 years. I was fortunate enough to spend many, many hours in our wetlands looking at beautiful habitat and these lovely aquatic plants. And you get nothing else from this presentation, want you to think, wow, those plants are really beautiful and they're really cool and an important part of these habitats. So I thought I'd show you this picture. Take a breath together, really internalize that idea. And now we can move into the research motivations. So for anyone who's unfamiliar, when I reference aquatic plants, these are plants that live out their life cycle either submerged or floating on the surface of the water. So in this image, that's really the surrounded by the yellow box. People will also refer to similar plant communities as things like macrophytes or submerged aquatic vegetation, SAV for short. And these plants are really important because of the ecosystem services they provide. One, these plants help to purify water. So their roots and structure help to retain sediment in the substrate. And they also are a really important part of nutrient cycling in our aquatic systems. Next, these plants are really important because they act as primary producers in our aquatic habitats. When you're out in the water, you're probably going to either have algae or aquatic plants. And I, for one, would much rather be hanging out with a bunch of beautiful aquatic plants than slimy algae. And then I would argue most importantly, these aquatic plants are really important because of the interactions they have with other species. That includes creating habitat and forage for waterfowl. They can be really important refuge for small fish trying to escape predators. And things like macroinvertebrates even lay their eggs on their stems and leaves. And there's really a great diversity in the shape, the form, the growth patterns of the aquatic plants that make up this plant community. So this screen actually has 15 different plants that I was able to observe myself out in our wetlands. And you can see some of these are floating species, some of them are fully submerged, some have big, beautiful pink flowers, others don't really flower. So there's a lot of diversity here, which is really important for the community's resilience. But unfortunately, like many things in natural resources, aquatic plants are currently being threatened by a loss of habitat, climate change, and invasive species. But unfortunately, aquatic plants are kind of being forgotten in aquatic restoration planning. So I would say aquatic restoration is really having a moment right now. People are excited to make our habitats healthier and happier. But unfortunately, we don't really have the resources to be doing that for our aquatic plant communities. So this was a paper actually published by our lab where we interviewed different vendors for wetland plants. And you can see that over half of the vendors had 0 aquatic plants available for people to purchase. So even if people are thinking about aquatic plants, which many aren't, and they want to be able to bring these plants back, They just don't have the physical resources to do so. And without those resources, our lovely bouquet of beautiful aquatic plants will slowly start to disappear. And we're gonna have less diversity and less abundance in these aquatic plant communities, which in result will lead to fewer ecosystem services, which we rely on. One of the things that's currently a big issue in North America is curly-leaf pondweed, or Potamogeton crispus. This is that invasive aquatic plant I mentioned, and that will really be the focus of the rest of this presentation. Curly-leaf pondweed is a common invader found throughout North America. It was originally native to parts of Europe, Asia, Africa, and Australia, but migrated here in the late 1800s, and you can see is really quite common in the northern Midwest, and just starting to encroach on areas of Utah. And like many invasive species, this plant will come in and it'll completely dominate a system. So this is a news article from Iowa where they called it a monster weed. It's taking over, creating a monoculture, and really making it difficult to access and use this waterway. However, there has been some research in short-term studies where they've actually shown curly-leaf pondweed isn't great at direct competition with some of our native aquatic plant species. So of course, that begs the question, how does curly-leaf pondweed become so successful in these systems? And how do we avoid those circumstances to help limit the invasion window of this problematic species? Okay, so to understand that a little bit better, we need some more background information on this plant species. So in contrast to many of our native aquatic plants, which will emerge in the spring, grow throughout the summer, and senesce in the fall, curly-leaf pondweed will actually acquire nutrients really early in the spring because it overwinters as small plants. So it's able to start growing sometimes when there's still ice on the surface of the water. But with this early growth also comes early senescence. So midsummer, when we're often already having a lot of issues with water quality in our waterways, this plant will senesce, die off, release a bunch of decaying biomass and nutrients into the water, which then encourages algae growth, which can also compete with our native aquatic plants. So this brings forth this idea of a lack of niche overlap between curly-leaf pondweed in our orange and our native aquatic plant species in blue, which kind of results in questions surrounding what the interactions will actually be between these different plants. How will the success of one species impact the other if they're not necessarily competing at the same time for the same resources? And then we complicate this even a little bit further because there are different phenologies or growth patterns of curly-leaf pondweed depending on where you are in the United States. So in the North Midwest, we have this kind of classic growth pattern that I've discussed here, but there have been some studies in the Southeast where they've shown curly-leaf pondweed growing and producing vegetative propagules throughout the year. So you never really have that overwintering population or that big senescence midsummer. Okay, and all this background is going to be really important as we go into chapter 2 of my thesis, which is looking at those interactions between curly-leaf pondweed and our native aquatic plants. But then, of course, we have to ask the question, what happens when curly-leaf is successful and is able to invade these systems? We have some examples of what native aquatic plants might look like, and we have an example of curly-leaf pondweed. Those are going to offer different structures for the fauna living in these habitats. So what will the impacts be on our wildlife? And we'll be talking about that a little bit with chapter 3 of my thesis, where we're going to explore the Junesucker habitat preferences. Okay, before we get deep into those research questions, I also want to introduce you to the study system I use to answer the questions. So all of this research was really inspired by the Provo River Delta Restoration Project. For those who aren't familiar, the Provo River is a tributary into Utah Lake, which I've highlighted in orange in that bottom map. And the Provo River, like many rivers in the United States, has become channelized as people develop the land around it. So you can see the remnants of this channelized river at the bottom. And this meant that we had really fast moving water and not great complex structure for different species trying to inhabit that area. And this had big impacts on Junesucker, which is kind of our fun fish of the day. Junesucker are a federally threatened species, and they're endemic to Utah Lake. So it's the only place you can find them in the entire world. And they rely heavily on these tributaries like Provo River for spawning habitat. And we've seen that these fish are able to go upstream and lay their eggs successfully, but we've also seen that there's this recruitment bottleneck for the young fish. We just don't see great survival of our larvae in juvenile. So right now we don't have a naturally recruiting population. So all of that meant that we needed to create some good nursery habitat. And that's what the restoration project is all about. This image is showing you the delta after restoration. So this whole part in the black and white line is the restored habitat. You can see really beautiful braided channels. We've got a good mixture of shallow and deep habitat. And the hope is that we're going to have some good aquatic vegetation in that area to provide that complex structure for small fish. Because we think the reason these small fish aren't surviving is actually predation. So we want to offer them some refuge habitat, some areas to find safety from these predators. And going back to our aquatic plants, two of the most common species I've seen at the Provo River Delta are sago pondweed and horn pondweed. So those are those top two. These are two really common aquatic plants throughout North America, as you can see with these maps, present in almost every state. And they have really complex structures. So fine leaves that are heavily dissected that occur throughout the water column. Okay, so for our study system, just to lay the story out there really plain, we've got these species of conservation concerns, the small June sucker, that are looking for habitat. We know from some surveys that there's an establishing native aquatic plant community, which is really promising. However, we also know that there's an upstream propagule source of curly-leaf pondweed, this invasive aquatic plant. So previous surveys have shown a thriving population of curly-leaf pondweed upstream in the Provo River, and we know that it's really likely this plant is going to migrate in and potentially have impacts on this restoration project and consequently the June sucker. So all of this together makes this a really ideal system for trying to parse these questions apart on relationships between our invasive species and our native species and how that might impact different wildlife like fish. Okay, I'm going to take a sip of my water and then we'll get started with the next section. So this next section is focused on chapter 2 of my thesis. This is where we're going to look at some of those relationships between curly-leaf pondweed and native aquatic plants. Remembering that we think there's this lack of niche overlap between the plant species, and we're really interested to know how the success of our invasive might impact our natives and vice versa. So for our first research question, we have, does the addition of native plant cuttings inhibit curly-leaf pondweed invasion and vice versa? Then we also wanted to be able to identify the phenology of curly-leaf pondweed grown at mid-latitude sites. Remember, I mentioned the phenology can be different depending on where in the US you are. And by identifying that phenology, we can hopefully take the research done in these other areas and start to apply those control methods to areas like Utah. Okay, and we worked to answer these questions via 2 studies. The first was a 16-month outdoor mesocosm experiment. And in this experiment, I added wetland soil from the Provo River Delta, so it had a propagule bank present, into these tanks in the summer of 2023. And then I added various combinations of our invasive plant and our native plants, and I let them grow. And then I continued to monitor them through the 2024 growing season, so that hopefully we're going to get some of those seasonal advantages or interactions between the plant species. For anyone who's unfamiliar, a propagule bank is just really soil that includes things like seeds, turions, tubers, or any other plant material that can emerge and help with an establishing aquatic plant community. For my second study, I did annual summer surveys of the curly-leaf pondweed invasion at the Provo River Delta. So I actually went out to the Provo River Delta, visited 15 randomly selected locations, and then at each of those locations, on my paddleboard, put my plot down and marked whether curly-leaf pondweed was present in that location. And that gave us some presence-absence data for the species. To analyze this data, we'll start with the mesocosm. I used a couple different generalized linear mixed effect models. So first off, I wanted to model the native aquatic plants. So we used the mean peak native plant cover, and we tried to model that with the fixed effects of whether that plant was added as a cutting and whether curly leaf pondweed was added. And we included the random effect of experimental block. Then we ran a pretty similar model for our curly leaf pondweed, where we were looking at mean peak curly leaf pondweed cover, and we used the fixed effect of native plant cuttings added. So either sago, horn pondweed, or no native plants added. And then we again included that random effect of experimental block. And both of these models were run on the 2024 growing season data. So we'll start with the horn pondweed. We'll build this graph out. On our y-axis, we have the peak mean horn pondweed cover. And then on the x-axis, we have whether that plant was added to the tank right here. And then each of our panels represent whether curly leaf pondweed was added, no, and yes. And we can see that there really wasn't a difference in our treatment applications. We think that the abundant propagule banks present in the Provo River Delta soil really overwhelmed any impact of our planting treatments. And remember, this is a season after the planting. So basically 1 year after we made the plantings, there wasn't really any impact of adding the additional plant material. We can say that is also true for sago pondweed. Again, no differences between those planting treatments. And one of the reasons that I think we're not seeing any difference between whether curly leaf pondweed was added or not is because curly leaf pondweed remained really low throughout the whole period of our experiment. So you can see the mean peak cover for those different treatments is never above 10%. It stayed really low. We think, or are hypothesizing, that this is because of the high native aquatic plant cover. Basically, curly leaf pondweed is being suppressed by all of our native aquatic plants. Just to give you some kind of qualitative evidence towards that, here are some photos from the beginning of the 2024 growing season. So this is April. And what we're seeing is in this tank, curly leaf pondweed was added both, but in this tank, we're seeing a lot of those overwintering curly leaf pondweed plants emerging and being healthy, successful. In comparison, we have in this tank decaying native aquatic plants and algae. And what actually happened is in this tank, we had really high cover by those native aquatic plants in 2023. So that material was still decaying, it was still there. Whereas in this tank, we never really got high cover by our native aquatic plants. So it wasn't there shading out the curly leaf pondweed propagules. So it seems like maybe there's something there, some sort of suppressive quality of the high cover in the previous growing season. To dig into this relationship a little bit further, we did one more model where we were modeling curly leaf pondweed cover for all of our data collection on 2024 by the cover of sago and horn pondweed and the planting treatment in that tank. And we can see that there's some weak evidence for lower curly leaf pondweed cover with higher sago pondweed cover. I know that this slope is pretty shallow, doesn't seem too exciting. I do think part of this might be that curly leaf pondweed was just never in high abundance, so it's hard to pull out kind of big slopes like that. But we can see that curly leaf pondweed cover was not really getting above that 20% mark unless sago pondweed was at nearly zero. Okay, now I just want to talk a little bit more about some of this qualitative assessment of our data. And we're going to transition to those surveys that were conducted at the Provo River Delta. So on our left, we have our 2023 data. On our right, we have the 2024 data. And the first thing I want to point out is that the occurrence of curly leaf pondweed actually didn't increase between the years. So we had the same number of subplots where curly leaf pondweed was present in both 2023 and 2024. The next thing I want you to notice is that curly leaf pondweed is spreading, right? So in 2023, we were a little scared. We saw a lot of curly leaf pondweed coming in where the Provo River enters. We were thinking there's a lot of propagules coming in. It seems to be successful. Maybe we're going to just see this density spread throughout the Delta. And we were really happy to see that that's not what happened. We actually had some really great populations of sago and horn pondweed here that seemed to be competing with the curly leaf pondweed. In contrast, we did see it spread, so it was in new areas of the Delta, but most of those areas were places where our native aquatic plant propagule bank just hadn't emerged, didn't seem to be as successful. So all of this is kind of pointing at that idea that abundant native aquatic plants might be able to suppress the ability of this invasive aquatic plant to really dominate a system. So we've kind of dealt with that first research question. Now I just want to show you one more graph from this study. And these are looking at the phenology of our 3 focal plants in our different plant treatments. So basically we have the proportional cover by our 3 different species represented by the 3 colors for our 2024 monitoring season. And I just want to kind of hone in over here on the horn pondweed and curly leaf pondweed. That's where we got the highest cover by curly leaf pondweed. And we can start to see that growth pattern that I mentioned earlier. Higher cover in the beginning of the spring, a midsummer senescence, and then we start to see some of those overwintering plants emerge. So that suggests we do have a growth pattern, maybe not as exaggerated, but similar to what's observed in the northern Midwest. And that's important because that means that we might be able to apply some of the management options that they use up there that rely on this phenology, including early season herbicide treatments before our native aquatic plants have fully emerged. Okay, so for our 2 research questions, first we can say, you know, the plants don't seem to be interacting or affecting each other in terms of our plantings, but we think that's because our native propagule banks were just so abundant that it really overwhelmed the planting treatments. And then second, when we're looking at the phenology of curly leaf pondweed, it does appear to be more similar to the northern Midwest. But what does this mean? What are the you know, considerations and implications for management? So we start with that idea that these native aquatic plants do appear to be limiting curly leaf pondweed invasion, which means that it's really important that we support these populations at restoration sites, particularly where we think curly leaf pondweed might be invading or coming. However, it's also important to recognize that propagule banks can play a really important role here. So if we know that we have abundant propagule banks at a site, it might be a better use of our limited resources for the restoration project to focus on other aspects. And then finally, that idea that our phenology is similar to the North Midwest, we might be able to start applying some of those control methods found in that area. Okay, now we're going to move on to my next thesis chapter, June sucker habitat preferences. And just as a reminder, this is coming to that idea where the structure is pretty different between some of our native aquatic plants and that invasive plant. And we're interested in how that might impact things like June sucker. And the reason we think this might be important is because in addition to some of those ecosystem services I mentioned earlier, aquatic plants are particularly important for small fish. They offer structure for orienting. They offer really good foraging grounds, including abundant macroinvertebrate and zooplankton populations. And then potentially the most important, they offer important predator refuge from things like non-native fish. So there's a really big white bass population. There's a growing northern pike population at Utah Lake. And we think that could potentially be a big issue for these small June sucker. And that all comes down to structure. So when we look at our 2 native species, we can see they're really complex. They've got a lot of divisions in their leaves, really fine. And then when we compare that to curly leaf pondweed with its broad leaves that also sometimes shade out growth in the lower canopy, we get a lot less structure for these fish to be using. Which brings us to our questions. Do June sucker exhibit preferences between historically available habitat and the invasive curly leaf pondweed? And do these preferences change when exposed to different predator stimuli? To answer this question, we tracked which habitat June sucker inhabited over an hour-long period when exposed to different predator cues. For our treatments, we used a full factorial design where we did every combination of group size. So either an individual June sucker or a cohort of 5 June sucker were added to the tank. And then every combination of predator treatment, which included a control, an avian predator, and a piscivorous fish predator. And then we did 10 replicates of each of these treatments. For our control, we just added the fish to the tank, let them go where they wanted to go. For the avian simulation, I had lovely volunteers like Karin swoop a gull decoy over the surface of the water. And we moved around the tank to make sure we weren't biasing one habitat. And then for our piscivorous fish simulation, we actually added a live white bass to the center of the tank in a transparent container that had holes drilled in it, so we could still get the chemical cues from the predator. And then because these were hatchery-reared June sucker that were predator naive, we also added in a con-specific alarm cue from the June sucker that told them there was danger around. Basically, it lets them know that there's another harmed June sucker in the water and they should be scared. And then for our response, we tracked the location of June sucker at 1-minute intervals throughout that hour-long observation period. We had a GoPro for the surface, taking a picture every second. So when we put those together, we were able to get kind of a video-esque result. I will say this is a little bit more grainy than the pictures I was looking at to get it into this format, but I just wanted to let you guys see what this ended up looking like. Small fish moving between habitats, selecting for where they thought they would be safest. And in these tanks, of course, we had the 3 different vegetation. We had the horn and the sago pondweed acting as our natives, and the curly leaf pondweed as our invasive. And then, of course, there's a lot of open space there as well. That's our sand. When you hear me talking about some of the results, you might hear me say historically available habitat. For me, that would include the horn pondweed, the sago pondweed, and the sand. Those have all been available to June sucker as they've evolved, right? Whereas curly leaf pondweed is a new habitat type for them. Okay, to analyze this data, I used 2 main methods. The first was a state transition model. And in this state transition model, we estimated parameters to create a transition matrix. And in this transition matrix, we have our starting locations. So sago, horned, curly leaf, and sand. And then we have the likelihood that it's going to end up in any of the 4 habitats in the next time step. So right here, these are the likelihoods that it would go from sago pondweed to any of the other habitats and itself. And we can see on the diagonal there, those bolded lines, those are the likelihood that the fish would stay in the habitat, which I might refer to as P stay or the probability of staying. One more example of that, if we had a fish that started in sand, we can then go to the sand column and pull those numbers and get the likelihood that it would be in any of those 4 habitats at the next time step. And this allows us to think about the movement of the fish and potentially model and simulate how fish will use these different habitats under various treatment combinations. To estimate these parameters, we used a Bayesian hierarchical state transition model, which I ran in JAGS or with JAGS in R. And we had a couple model assumptions we had to use in order to estimate our various parameters. First, we assumed that the likelihood of staying in a habitat, so those diagonal numbers, would vary depending on the perceived quality of that habitat by the June sucker. Next, we assume that perceived quality would also influence the likelihood of fish entering that habitat from another habitat. And we used a term called, relative value, basically how nice do June sucker think this habitat is. And we assume that those would also differ between the habitat types. And then finally, we assumed that the preferences between these habitats would depend on, basically the different predator treatments or group size that were applied to the fish. So our treatments would have an effect on their movement. Okay, all of my graphs going forward are kind of going to be in a similar setup, so I just wanted to introduce you to this. For our columns, we have the 3 different predator treatments. So we have control, avian, piscivorous fish. And then for our rows, we have cohorts of June sucker on top and individual June sucker on the bottom. Okay, now I'm going to flip the screen. You're going to see a lot of numbers. It's a little overwhelming. Don't worry about it. I just want you to look at these diagonal numbers, the ones I've highlighted in blue, and realize that those are all pretty high. Most of them are above 0.9. And that basically means that June sucker were really likely to stay in the same habitat they were seen in the previous time step. They weren't moving around a whole lot. Next, we can use these transition matrices to actually simulate movement of June sucker. And if we look at the bottom row for our individual fish, we can see that our observed and simulated data are pretty similar. We're falling within those standard errors bars for our observed data. Unfortunately, when we go to our cohort data, not quite the same story. We can see some areas where our model really is either overestimating or underestimating the use of a habitat. And we think that could be for a couple different reasons. First, we were trying to model individual movement with cohort level data. So basically at each time step, we just had the count of fish in the habitats. We didn't have an individual fish location for those cohorts. And we also weren't able to incorporate some of the behavioral aspects of these fish being in cohorts. Maybe fish were competing for the same habitat and pushing each other out, or they were moving as a school and traveling together. We don't really know exactly what we're missing, but we're definitely, not quite hitting the mark with that cohort model. So from here on out, for this specific transition model, I am going to focus on the individuals. Okay, so what we were really trying to get at with this study was, is curly-leaf pondweed an equivalent quality habitat for June sucker? So in this graph, I've actually subtracted the P stay values of curly-leaf pondweed from the P stay values of our historically available habitat. And if we have a number close to 0, that basically means there wasn't a big difference in that perceived quality of the habitat. So we can see for the control and avian, close to 0, not a whole lot of differences. But then we move to the pestiferous fish, huge differences, right? We have all 3 of our historically available habitats are being preferred and used more than our invasive curly-leaf pondweed, presumably because they don't see it acting as a high-quality habitat or refuge from these. And we can see a similar trend if we look at those relative values or the likelihood of a fish moving from one habitat into curly-leaf pondweed. So we can see that the likelihood of moving from sand, horned, or sago pondweed into the curly-leaf pondweed was very low for the piscivorous fish treatments. Basically, they didn't want to stay in there, and they also didn't want to go in there. Okay, next we're going to move on to our second model for this chapter, another generalized linear mixed effect model. And in this model, we were trying to assess habitat selection, so which of those 4 habitats a fish is going to, using the fixed effects of group size, either cohort or individual, and predator treatment, control, avian, or piscivorous fish. And again, we're including that random effect of experimental tank. Okay, same setup as before, predator treatments as columns, fish group size as rows, and we're going to start with our control. And we can see that there's a slightly lower use of sago pondweed for both the cohorts and individual fish. It's still being used, but not as much. I think this is really important to keep in mind because sago pondweed is a native plant often targeted for restoration because it's really seen as a high value plant for waterfowl, but obviously maybe not a high value plant for these fish. So it's important to keep in mind multiple wildlife species as you're making these decisions. Next, we can move to our avian treatment. We can see even fewer differences in the use of these habitats. They're really all being selected for at different times by different fish. I do, however, want to point out that the use of sand for the avian treatment is much lower than it was for the other 2 predator treatments. Basically, if a June sucker saw something swooping over the surface of the water, it didn't want to be out in the open. It wanted to find cover and be able to hide from that potential threat. Okay, and then we can move to our piscivorous fish treatment. And the results here are really emphasizing or backing up what we saw in that transition model, right? Really low use of curly-leaf pondweed, particularly for individual fish, and also pretty low use of curly-leaf pondweed for the cohorts of fish. I can say in the hundreds of photos I looked at of June sucker in this treatment, there were only 3 that actually had an individual June sucker using curly-leaf pondweed. So for whatever reason, they really didn't see that plant as a good habitat when they were threatened by a large piscivorous fish. Okay, so then if we return to our research questions, we can say, yes, June sucker exhibit preferences. There were some differences there. And while June sucker use a variety of habitats, they don't often utilize curly-leaf pondweed when exposed to those piscivorous fish predators. Moving into some management implications. So knowing that they exhibit these different preferences depending on various predator simulations, we can kind of come to the conclusion that June sucker are going to need a variety of habitats, including complex structure provided by some of those native plants and open space, right? We did see sand used. Sometimes fish, rather than hiding, might want to try to escape. Maybe that isn't the best idea, but they're going to try it. And then we can also come back to that idea that curly-leaf pondweed was really not perceived as good refuge habitat, specifically when these piscivorous fish were present. And that curly-leaf pondweed might need more management at the Provo River Delta, depending on how its invasion progresses in the future. So it'll be important to continue to monitor that. Okay, next we're going to move into just some final thoughts and considerations for aquatic plant management. So as any scientist, I was wrapping up my thesis and I started thinking, oh God, is anyone going to use this work? You know, I want this to be applied. I want people to be able to access all this knowledge and experience that I've built over the past 2.5 years. And that led me to creating this one-page graphic with recommendations for aquatic plant management and restoration. Hoping this will be useful for people across North America, but I was definitely coming at it from the lens of the Intermountain West. But I would be remiss if I didn't mention that this wasn't just me pulling from my own research, but also pulling from conversations, interactions I've had with other people in our lab. So starting with Kate Sinnott, she was a master's student in our lab that focused on different planting techniques for aquatic plants. Sandy Johnston continued to work on this idea of different plant treatments for different aquatic plant species. I've also had two really amazing undergraduate researchers, Chloe Francis and Ashlyn Monroe. And then of course, Sam Kurkowski, our amazing lab manager, who somehow has her hands in every project happening. So I won't go into the details of this entire document, but I just wanted to highlight a couple of the sections that pulled from my research that I just discussed. So if we start with that assess section, basically I want managers to go out and understand what they already have at their site. And an important part of this is evaluating the propagule bank. And we saw that when we were looking at our mesocosm study results, which suggested that if you have abundant enough propagule banks at your restoration site, you might not need additional plant cuttings. And that those resources could be better used on other aspects of your restoration project. Next, it's really important to assess potential upstream propagule sources. Of course, this is referencing that curly-leaf pondweed that was upstream in the Provo River Delta that we were aware of and able to monitor its invasion progression through the past couple of years. And that's been a really important aspect of knowing if we need to start worrying about control methods, what kind of impacts it's having on the ecosystem as a whole. Next, we get into project goals. This is where I suggest people consider how aquatic plants might actually influence or help along the success of various project goals. And for this one, I'm pulling from some of my information on the June sucker chapter. Basically, knowing that June sucker have preferences for specific plants or habitat types means that we should be trying to restore those specific habitat types or managing against others like curly-leaf pondweed. Then we get to supportive actions. One of the supportive actions I suggest people take is manage for invasive species. The thing that I think was most applied here from my own research is this idea that curly-leaf pondweed phenology at our mid-latitude sites is pretty similar to that in the northern Midwest. If you remember that range map of curly-leaf pondweed, there's a lot of curly-leaf pondweed up there. They've done a lot of really great research on control methods. Knowing that we have a similar phenology means that we can try to apply some of those methods here. So things like those early herbicide treatments before native aquatic plants emerge, or mechanical removal of small overwintering plants before they have their propagules set. And then like any good scientist, I had to include monitoring. We of course need to monitor our sites that try to assess how our management actions are working and adjust from there. Additionally, this information will be really important for future restoration projects. So for this, I want to highlight those annual vegetation surveys we did at the Provo River Delta. This has given us hope, right? We can see that curly-leaf pondweed seems limited by our native aquatic plants, but of course we need to continue to monitor. This is a master's project. It was only 2 years. We don't know what that progression is going to be like into the future. So monitor, monitor, monitor. Okay, that's all I have, except for some quick thank yous. I want to start with the June suckers. Thank you to the June suckers for making research possible. And thank all my funders and collaborators, specifically Dr. Timothy Wellsworth and Morella. You guys have been a really amazing committee. Hopefully I'll still say that in 3 hours. Also, I really want to give a huge thank you to Chad Teal and Phil and Andrew. I came to Chad maybe like a month after he started, and I said, I want to do an experiment in your new lab that isn't set up yet. And he said yes. And Phil and Andrew were amazing as I was sorting out all the logistics for my experiment. I think, Phil, my most memorable moment is you eating a jerky stick as you carry PVC pipe over your shoulder. You guys really saved me a couple times there, so thank you. I also want to thank Kelvin. He's the interim stats person for the Ecology Center. He was really helpful as I was putting together some of these models. Thank you to some of my Utah DNR folks, Gary Howe at the hatchery and Andrew and Keith, really helpful with in the field logistics, acquiring white bass for my experiment. And then thank you to the folks at the Provo River Delta, including Melissa Stamp, Paula Bate, and Eric McCauley. And then, of course, thank you to Karin for being such an amazing mentor. She really is a powerhouse. I'm always impressed by how much she's getting done. But she also always reminds us that we need to be whole people and do other things out of science. And so this is a picture of her and her husband, Mike, playing some Irish music. I really so appreciated that you were encouraging of having a life outside of school. I think so important. And of course, Karin also does a really amazing job cultivating a great lab community. So thank you to all the previous and current members of the Kettering Lab. And also all of the previous communities I've experienced in my various scientific roles. I think those cover most of the places Karin mentioned at the beginning of the presentation. Thank you to all my friends and the wonderful people in the Watershed Sciences Lab. Thank all of you for coming today. A huge thank you to my cool dog, Pepper, and my amazing partner, Julia. She has really been the most supportive person I could ask for throughout this whole process. And then thank you to all my family and friends, a lot of which I know are tuning in via Zoom right now. So with that, take questions. Yes, Chuck.
Hey, Megan, interesting study. So I have a couple of questions. Mesocosm experiments are susceptible to artifact. So how well do you think the mesocosm environment out at Millville represents the Provo Delta, i.e. are temperatures the same, et cetera?
Yeah, that's a great question. That's definitely a concern, right? We're never going to be able to create the same exact conditions. I think what we were able to achieve in the mesocosms resembles what we see in field sites, maybe not exactly the Provo River Delta, but other sites throughout Utah. We obviously collected soil from a local wetland. We made sure to have water running through those tanks every hour for 10 minutes. So we had water flow. We also used reef power heads to create circulation in the water. And we were really wanting to have this be an outside experiment to try to get at some of those seasonal interactions as well.
So the second question is, I suspect June sucker aren't the most intelligent animals.
Heart is breaking up here.
Reasonable prey would prefer to hang out in sand, say, come and eat me, versus heading for whatever cover might be around. And what is it about your study plant that keeps that sucker from going in? Is it just so dense they can get in, or what?
Yeah, so it's a great question. I think a couple things there. One thing I didn't talk about is in these experiments, we're really seeing kind of two separate behaviors by June sucker. Most of them were looking for refuge in vegetation. So I didn't show this graph, but in all 3 of the predator treatments, about 75% of the fish were using vegetation of some sort throughout the experiment. Then we had a smaller group of June sucker who seemed convinced that they could outrun the predators. And they would swim laps around the tanks. And that's why you were seeing higher use of sand, specifically during the piscivorous fish treatment. I think the conspecific alarm cue really freaked some of the fish out. which is the goal, right? Some fish are probably gonna do that out in the wild too.
Thanks.
Yeah, Wayne?
Yeah, a related question, and I was surprised by those fish out in the sand too. Are those fish from a raceway where they haven't had natural experiences? Are they looking for open cover like they were raised in, or are they fish that we're seeing from the lake?
Yeah, those were hatchery reared fish. In the previous summer, it actually attempted to do some surveys with DNR looking at vegetative use of small June sucker in the lake. I think they caught a total of like 20 fish, so not very many. They were having a hard time finding them. So we had to use the hatchery reared fish as our best proxy. So it's definitely a consideration. These fish are not necessarily the young fish you're gonna see out in the Delta, but it's kind of the best information we can get at right now. That was also why I included that con-specific alarm cue. These fish obviously haven't been exposed to predators in the past. So I wanted to make sure there was some sort of, you know, innate response there, that there was a threat that they should be scared of. Casey.
Great job, Megan. My question kind of hinges on one thing that I kind of forgot. Did you mention the white bass was invasive as well?
The white bass is a non-native sport fish.
So, you know, from one side of it, as someone who is not, you know, studying fish or plants, you got that highest response from the white bass or the predatory fish. So in your opinion, you know, what is a more effective method? Is it removing these invasive fish species or thinking of the plants alone? Because, you know, those obviously drive that, and so you've got these two kind of interacting elements there.
Yeah, great question. I think as much of restoration science, I would say you should really come at it from both sides, right? So maybe we should be taking efforts to control those white bass populations. There's also competing things there, right? People like to sport fish white bass, they're already present. And maybe we should take some steps in addition to also control the curly leaf pondweed population. Come at it from both sides and hopefully have some success. Yeah.
Do you have a sense of why curly leaf pondweed is so successful in the places where it has been successful? Like, I'm guessing there are probably native plants there, but you're not seeing that competition, or like, is it just once it starts, it just goes, I guess?
Yeah, that's a great question. I can speculate. I think there are a couple things. I would say aquatic plants are kind of the ugly ducklings of wetlands, and people don't really like aquatic plants. And aquatic plants in general have been on the decline. So I think there are a lot of disturbed habitats, aquatic habitats, where propagules for curly leaf pondweed are coming in, and we just don't have this healthy, abundant native aquatic plant population to be competing with it. I also think those seasonal advantages of the early acquisition of resources are really important to that dynamic. Just hoping, what I was hoping to capture in the mesocosm experiment, but it just didn't take off like we were expecting. Yeah, so complex story. I think there's a couple pieces there. Yeah, Ed?
Yeah, harking back to mesocosm study, it looked like in your change of state model, sorry, I forgot what you called it exactly. Yeah.
Okay.
But it looked like from no matter where you were, the most likely place you were going to move to was sand. Part of that might be the proportion of that mesocosm that is covered by sand.
Right.
So if you just, did you think about, I can put that proportional cover in and actually generate expected, where I'd expect to find the fish was totally random versus where I did find the fish based on the proportional covers?
I did think about that. Kind of the conclusion I came to is fish out in the wild are never going to be exposed to perfectly equal amounts of habitat. So I was really more interested in the general trends than those exact numbers. So, you know, the trend that curly leaf pondweed is used a lot less during the piscivorous fish treatment, I think would obviously or hopefully still be present if there were completely equivalent uses of those habitat. Yeah, but good question.
I think it might actually exaggerate.
I think it would too, yeah. Yeah, Greg.
You said in your introduction, like you point out, that the Provo River Delta is highly altered from its historic state. I'm wondering, with your recommendations for managers, are you treating, or do you think that the Delta is in a fairly stable state as it is, or do you think these recommendations should incorporate other potential changes?
Yeah, do you mean the graphic at the end or some of those recommendations that I made throughout?
More throughout.
Okay. Yeah, I think a big part of those recommendations throughout is that last thing I just talked about with the monitoring. It'll be important to see how the curly-leaf pondweed invasion progresses. Right now, I think managers need to be aware of the situation and ready to start control methods if needed. It seems like for now our native aquatic plants are handling themselves, but definitely having adaptive management in these areas will be really important for the long-term success of the restoration project.
And so I actually have two questions. My first one is this year, the smartweed in the Delta had a fantastic year. It absolutely blew up.
Yeah.
Especially on the north side. I'm on the side of assuming that the utilization of June suckers, should they be in the Delta, would be to utilize the smartweed much like they would sago or horned. Would you agree with that assumption, or do you think that the morphology of smartweed might have a different response from the June suckers? What's your thoughts with that?
I would actually say water smartweed might be a little bit more similar to curly leaf pondweed, right? It has those broad leaves, and it also doesn't have a lot of structure throughout the water column. You're seeing more floating leaves. So I think it could be a potential great resource for coverage from aerial predators, right? But we still will need things like sago and horned pondweed for some of those predators like white bass.
Interesting. Okay. And then my second question, and it probably just has to do with time and that sort of thing, but I'm curious, what would your thoughts on if you started a plot with a high amount of curly-leaf pondweed and then seeing if the natives are able to make it through, just looking forward in time, say if is it a function of just how long we've been observing the system, or maybe once curly-leaf reaches a critical level, it's hard to come from, is kind of what I'm asking about.
Yeah, I think that would be a really interesting study that could definitely add some information to what I've been able to gather from my research. We were interested in that kind of prevention and early detection. Something like that study where you already had a really abundant population of curly-leaf pondweed is probably going to require a more aggressive control method like herbicides.
Interesting, thank you.
Yeah. Any questions on Zoom?
No, just a bunch of congratulations.
Okay, well, let's thank Megan for.
Very ready. It'll be. Yeah, thank you, Jeff. I'm for ready. I know. Yeah, so I actually the other girl who and that was on there. I students and they're like, well, they probably know what you're talking about. So yeah, I actually did not have the internet, but my friends and yeah, good, I'm glad. I feel like yeah. I did 16. Yeah. Barely.